Hello. Today I'm going to be talking about how CRISPR is being used to potentially help treat cancer. Specifically, I'll be talking about the first clinical trial in the United States that tested a CRISPR made cancer therapy. So, to start things off, what is CRISPR? CRISPR, or clustered regularly interspaced short palindromic repeats, are segments of DNA containing short repetitions of base sequences. They were originally found as a component of prokaryotes' immune system, protecting against viruses, but they have been repurposed as a gene editing tool, and that's what CRISPR is known for. Scientists use a CRISPR sequence of DNA and its associated protein to edit the base pairs of a gene. So, how does CRISPR work? Well, first, the Cas9 protein forms a complex with an accompanying bit of RNA called a guide RNA. Then this complex joins itself with a matching DNA sequence next to what's called a spacer, which is in yellow in this picture. The Cas9 RNA complex cuts the double stranded DNA so that now a researcher or scientist could insert their gene of interest or strand of DNA there. Although CRISPR Cas9 has many advantages, there are some limitations that have been observed. The first one is sometimes it will cut DNA outside of the target gene, which is called off target editing. This could be harmful and might even turn cells cancerous, which is the opposite of the goal here. Another downside is that delivering CRISPR components to certain organs or cells in the human body can get complicated. Typically, to do this, a virus is altered to carry genes for the guide RNA and Cas is used. Some of these viruses can affect more than one kind of cell, meaning it could edit something like bone cells when it was intended to edit pancreas cells. Another concern is how the human body's immune system will react to these viruses carrying CRISPR, or just the CRISPR components alone. Some scientists and researchers worry that the immune system could attack Cas and destroy CRISPR edited cells. There is research going on today to learn about and hopefully fix these potential roadblocks. Okay, so now getting into CRISPR Cas9 and the possibility of it being used as a form of cancer treatment. I'll be talking about the research done at the University of Pennsylvania and the first clinical trial done there, well, and also just in the United States in general, in 2019 that tested this type of cancer treatment. They tested a type of immunotherapy where the patient's own immune cells were collected, genetically modified to better seek out and kill cancer cells, then infused back into the patients. This involved making 4 genetic modifications to T cells, which are immune cells that kill cancer, by the way. The first one is adding an artificial gene that gives those T cells receptors that can seek out a molecule on some cancer cells that's called NY-ESO1. The other 3 modifications are the deletion of 3 genes, 1 that hinders the cell's ability to kill cancer, and 2 that interfere with the NY-ESO1 receptors. So there were 3 patients involved in this clinical trial. One of them had metastatic sarcoma, and the other 2 had advanced multiple myeloma. But one thing that I should point out here is that all 3 of the patients' tumors contained NY-ESO1, which is important because that is the target of this therapy. The main goal of this clinical trial was to determine if CRISPR-made treatment was safe, although of course another big goal is to see if it was effective. Okay, so now getting into the actual procedure that was done, let's go through the steps. So first, blood was taken from the patient so that their T cells could be obtained. Next, they inserted the gene for the NY-ESO1 receptor and deleted the 3 other genes, which were PDC1, TRAC, and TRBC, from the T cells using CRISPR. Then they grew millions of those CRISPR-edited T cells and infused them into the patients. And the goal was for the edited T cells to bind to cancer cells and destroy and kill them. So getting to the results, there was no evidence of harm from this treatment, which was good. There were a few side effects, but it was likely from the chemotherapy that the patients received before their infusions. There was no evidence of an immune reaction to the CRISPR-edited cells, although only about 10% of the T cells used for the therapy had all 4 of the desired genetic edits. One bad thing is that off-target edits were found in the modified cells of all 3 patients, but none of those cells grew in a way that indicated they had become cancerous, which is good. Overall, the treatment had a small effect on the patient's cancers. Two patients' tumors, one with multiple myeloma and one with sarcoma, stopped growing for a while but started again after some time. Then for the third patient, the treatment just didn't work at all. The doctor said that even though there wasn't much effect on the cancer from the edited cells, the fact that the edited cells were able to temporarily stop the growth in the sarcoma patient was notable because it had been difficult to deal with solid tumors using cell therapy. This study marked the first trial of a CRISPR-made cancer treatment, in the US, but many more have been done since this one. CRISPR methods are still being improved, and hopefully that will continue to be the case. Thank you.